Okay, in the last class we had discussed on the I had given you the introduction on carbohydrates. So we are going to study in detail about the structure and class of and different class of carbohydrate molecules. So before we are going to discuss in detail about that, let us see about the stereochemistry. You need to understand about the stereochemistry of carbohydrates. Okay. So in this video, I'm going to talk about the stereochemistry. Now, what are isomers? As you have already studied in chemistry, also in chemistry class. So here I'm not going to discuss so much in detail, but just to remind you before we are going to discuss in detail about the structure and function of different class of carbohydrates, let us just revise, okay, about all these different stereoisomers, okay, stereochemistry of carbohydrates. So let us talk about this topic here, that is isomers. Now, what are isomers? We already know that isomers are those substances with the same molecular formula, okay, but with different arrangement of the atoms in space. So they have same formula, but they differ from each other only in the arrangement of their atoms. So those substances are referred to as isomers, and it is mainly of two types, that is structural isomers and stereoisomers. So if we talk about the first one here, that is the structural isomers. Structural, why they are referred to as structural isomers? Because they actually have same structural formula, okay, but they differ in their molecular structures. That means their molecular structures are different. So again, on this category, that is structural isomers, we have chain isomers, then we have positional isomers and functional isomers. All these three, they fall under this category, that is structural isomers. Now coming to the first one, that is chain isomers, here as you can see that they have different arrangement of the carbon atoms. For example, if we talk about butane, you can see here the formula is C4H10. But what is the difference between the N-butane and isobutane is just in the arrangement of the atoms. You can see clearly in both these substances, that is N-butane and isobutane, they are nothing but they are chain isomers, as they differ from each other in the arrangement of their carbon atoms. Now the next type here, that is the positional isomers. So positional isomers, they are structural isomers that can be viewed as differing only in the position of a functional group, okay, so only in the position of the functional group or substituent or some other feature on a parent structure. So as you can see here clearly in these two molecules, that is propyl chloride and isopropyl chloride, they have the same formula, okay. The formula for both these two is C3H7Cl, but you can see the position of the functional group here is totally different in these two substances. So these are the examples of positional isomer where the position of the functional group is different, okay. And the next type of isomers are functional isomers. Now functional isomers, again, they have the same molecular formula. For example, in N-propanol and methyl ethyl ether, they have the same formula C3H8O, okay, that is the formula of these two isomers, but they differ in their functional groups. Okay, so you can see the functional groups in these two isomers is different. So don't get confused in between these two. These, they have the same functional group, but the position of the functional group is different. So that is why they are referred to as positional isomers. Whereas these two, they have same molecular formula, but the functional groups are different. Alright, so all these three, that is functional isomers, chain isomers and positional isomers, they are referred to as structural isomers. Now we come to the next group, that is stereoisomers. Now stereoisomers, they have same molecular formula as well as same structural formula. Okay, that is why they are different from structural isomers, because they have the same structural formula. But what is the difference? They differ only in the configuration. Alright, only the configuration is different, that is the arrangement of their atoms in space. Now again, under this category, we have the first one, that is geometrical isomers. Now in geometrical isomers, what happened here? The one of the isomers has functional group on the same side, and the other isomer, it has the functional group on the other side. As you can see here, fumaric acid, it has functional group on the opposite side, whereas malic acid, it has a functional group on the same side. So these type of compounds or substances or isomers, they are referred to as geometrical isomers. Remember, they have the same molecular formula and same structural formula, but the difference is only in the arrangement of the atoms. For example, in case of geometrical isomers, where we have the functional groups are on the same side or they are on opposite side with respect to the double bond here. You can compare with respect to the double bond. You can see in case of malic acid, they are on the same side, whereas in fumaric acid, they are on the opposite side. Alright. So the next category here, we have the optical isomers. Okay, sorry, this one, those which have on the same side, they are referred to as cis configuration. Okay, this configuration is referred to as cis, whereas this configuration where the functional groups are on the opposite side is referred to as trans. Okay, trans configuration. So coming to the optical isomers, actually these isomers, they have again the same formula and functional groups, but again they differ in the arrangement of functional groups in space. Okay, so the arrangement of functional groups in space are different, and actually such type of isomerism is to the presence of a particular carbon atom, which is referred to as an asymmetric carbon or a chiral carbon atom in the molecule. Alright, due to the presence of this chiral carbon atom. Now, a chiral carbon atom is that carbon atom where it is attached to 4 different types of atoms or groups. Okay, so a carbon atom which is attached to 4 different groups of atoms is known as asymmetric or chiral carbon atom. We will talk about this in detail. So I hope it's clear. These optical isomers, they, due to the presence of these chiral carbon atom in their molecule, they possess this isomerism. Okay, they can form optical isomers. They have same molecular formula and functional groups, but just the arrangement of functional groups is different. Okay, the functional group are arranged in a different way in their atom, in their molecule. So by virtue of the presence of this chiral or asymmetric carbon atom, the molecule, it usually becomes optically active. When I say optically active, it means that these molecules, they can rotate in the plane polarized light. Okay, they can rotate the plane polarized light in either clockwise direction, which is indicated by a plus sign, or in an anti-clockwise direction indicated by a negative sign. Okay, simple substances which show optical isomerism, they can exist as two isomers known as enantiomers. Okay, so one more term you have to remember here, enantiomers. So those substances which can either rotate the plane polarized right in either clockwise or anti-clockwise direction, they exist as two isomers known as enantiomers, and these enantiomers, they are structurally superimposable mirror images of each other. Okay, so these molecules, these two isomers, they are mirror image of each other. Now, for example, you can see here when a beam of light, when a beam of polarized light is passed through a solution, all right, you can see here this is a beam of light, and this unpolarized light, it vibrates in all direction, in all planes. But when passed through a polarizer, okay, this plane of polarized light, when it is passed through this optically active solution, for example, here in this solution, it contains both these isomers, that is the enantiomers. Alright, when these enantiomers are present here, they will either rotate the light in a clockwise direction or anti-clockwise direction. Okay, so when a beam of polarized light is passed through a solution here, exhibiting optical activity, it will be rotated to the right or to the left, okay, in accordance with the type of compounds that is present here. That is optical isomer which is present. It depends on the optical isomer which is present in this solution. It will rotate the polarized light in either clockwise or anti-clockwise direction. Okay, so a compound which causes the rotation of the polarized light to the right, all right, if it rotates the polarized light to the right, it is said to be dextrorotatory. Let me show you. It is said to be dextrorotatory if it rotates the polarized light to the right, and is usually indicated by a plus sign and a small d. Okay, small letter d, and levorotatory, small letter L, when the polarized light is rotated to the left. Okay, and is usually designated by a minus sign. So if you see this in the names of any compounds, if you see this positive or negative sign or a small letter D and a small letter L, it just means that this compound, this isomer, it rotates the polarized light to the right if it's a plus sign and to the left if it's a minus sign. Okay, so here when equal amounts of both the dextrorotatory and levorotatory isomers, they are present, okay, when equal amounts of these two are present, the resulting mixture, it has no optical activity. Why? Since the activities of each of these isomers, they cancel each other. Okay, such a mixture where equal amount of dextrorotatory and levorotatory isomers are present, that particular mixture is said to be a race mix mixture or DL mixture. Okay, D for dextrorotatory and L for levorotatory, and thus a compound, it might be designated as plus or with a minus sign for levorotatory and a positive sign for dextrorotatory compound or isomer. So the next topic that we will discuss is on DNL sugars. Now these D and L sugars I had mentioned just now about the enantiomers. Okay, enantiomers, they are simple substances which show optical isomerism. And these simple substances which show optical isomerism, they exist as two isomers known as enantiomers. So here D and L sugars or carbohydrate molecules, they are also termed as enantiomers, and their structures, they are mirror image of each other. Now, why these enantiomers are named as D and L form, it depends upon the arrangement of the hydrogen and hydroxyl group on the pre-last carbon atom, okay, which is also the last asymmetric carbon atom. Here, asymmetric carbon atom or chiral carbon atom, I had already mentioned, it is a carbon atom which is attached to 4 different groups. That is referred to as the asymmetric carbon atom. Now, the pre-last carbon in a molecule, okay, in a molecule, the pre-last carbon or the asymmetric carbon atom, you have to refer to that particular carbon atom, and when the arrangement of hydrogen and hydroxyl, it depends on the arrangement of these two on the pre-last chiral carbon atom or asymmetric carbon atom. That will be known as D or L form. So here you can see that when the sugar, it has a hydrogen group on the left and a hydroxyl group on the right of the penultimate carbon atom, that is the pre-last carbon atom. Alright, that form will be referred to as the D form. Whereas the opposite arrangement, that is when the hydrogen is on the right and hydroxyl group is on the left, that arrangement, in that case it will be referred as the L form. Alright, but many naturally occurring sugars, they usually exist as D isomers, indicated by D and capital D and capital letter L. Okay, so just remember that here small letter D and small letter L, it indicates the rotation of polarized light, whether to the right or to the left, also indicated by a plus and a minus sign. But here this capital D and capital L, it indicates, it depends on the arrangement of these two on the penultimate carbon atom. Okay, if the hydrogen is on the left and hydroxyl on the right, it is D form, and if it is vice versa or opposite arrangement, it will be termed as the L form. So that you will see many examples in later on when we will study about the detailed structure. So for example, here in this case, you can see this is referred to as D-glyceride. Why? If you see here, this is the chiral carbon atom. Why it is the chiral carbon atom? Because it is attached to 4 different groups. This one, the hydrogen, the OH group, and this group. So there are 4 different groups attached to this chiral carbon atom. And why it is a D form? Because hydrogen is on the left and hydroxyl is on the right. But this one is the L-glyceraldehyde. Why L here? L, it indicates, if you see the arrangement here, the hydroxyl is on the left and the hydrogen is on the right. So these are the examples of D and L sugars, also known as the enantiomers. Same thing here, if you see in case of glucose, we have 4 asymmetric carbon atom. Alright, these 4, 1, 2, 3, 4. If you take a look at the first asymmetric carbon atom, you can see why it is a chiral carbon atom. This one, you can see it is attached to this group, the OH group, the hydrogen group, and this group. So this carbon atom is known as chiral carbon atom. Same thing if you see here, this one, it is attached to this group here, this OH group, hydrogen, and this whole group. So it is attached to 4 different groups here. This is the second asymmetric carbon atom. So if you take a look at this carbon atom here, you can see it is the pre-last carbon atom. Alright, it is next to this. So this pre-last carbon atom, you can see that the hydroxyl group is on the right. That is why it is D-glucose. Whereas in this case, if you take a look at this pre-last carbon atom here, the OH group is on the left, so it is L-glucose. So I hope it is clear about the D and the L configuration, okay? DNL sugars. Now, I just want to show you this glyceraldehyde, that they are mirror image of each other. As you can see here, glyceraldehyde, it is the simplest carbohydrate, and it exhibits the properties of a chiral or optical isomer compound. This glyceraldehyde, you can see it can exist in 2 isomeric form that are mirror images of each other. Okay, as you can see clearly in this figure, that these are the 2 isomeric form of glyceraldehyde, that is D and L, which are mirror images of each other. And another example here is this one, the D-glucose. Alright, the glucose molecule. Another example of enantiomers is the D and L isomers of glucose molecule, as you can see in the penultimate carbon atom, where the OH group is on in this isomer, the OH group is on the left, that is D-glucose, whereas in this isomer, the OH group is on the, here it is on the right and here it is on the left. So these are the 2 isomeric form of glucose, or also known as enantiomers. So the next topic that we will discuss, that we will talk about, is epimers. Now, epimers, they are nothing but they are isomers which differs in the configuration at only a single carbon atom. Alright, that means all other carbon atom, they are similar. The arrangement of the atoms, they are similar, but they differ from each other only at a single carbon atom. For example, if you take a look at glucose and mannose here, what happened in glucose and mannose, if you take a look, everything is the same from carbon number 1 till number 6. It's the same except in carbon number 2, where in carbon number 2 in glucose, the OH group is on the right, the hydrogen is on the left. Whereas in mannose, the OH group is on the left and the hydrogen is on the right. So the difference is just at a single carbon atom, that is carbon number 2. So these are referred to as the C2 epimers. Here it indicates that they are epimers. They differ from each other only at carbon number 2. Alright. Similarly, if you compare glucose and galactose, they are C4 epimers, meaning they differ in conformation only at the 4th carbon atom. Everything else is the same except carbon number 4. And these epimers, they are also termed as diastereomers. Okay, diastereomers. They are also referred to as diastereomers. And the interconversion of epimers, for example, glucose to galactose, or galactose to glucose, okay, or vice versa, that process is termed as epimerization reaction, and a group of enzyme epimerases catalyzes the reaction. Okay, so the interconversion of the epimers, that is from example from glucose to galactose or galactose to glucose, that is termed as epimerization reaction, and a group of enzymes called epimerases catalyzes this particular reaction. So that is about the epimers. Now we come to the next term, that is anomers. Now, in solution, the sugars or the carbohydrates, they exist as ring structures, okay, and not as straight chain. That I had already mentioned earlier in my previous video, that in solution they actually exist in ring structures, and there are actually two different ring forms, that is alpha and beta. These alpha and beta they form as a result of hemiacetal formation between the hydroxyl group of the fifth carbon atom and the CHO group of the first carbon atom. Alright, and these two they are termed as anomers, and this first carbon atom, alright, since you see that the hemiacetal formation, it occurs between the OH group of the fifth carbon and the CHO group of the first carbon atom. So this first carbon here is termed as the anomeric carbon atom. Anomeric carbon, and the two forms, okay, the alpha and the beta forms, which results due to the hemiacetal or hemiketel formation, those are termed as anomers. Now, if the OH group on carbon 1 and CH2OH group on carbon 5 are on the opposite side, alright, when this structure is formed, okay, so when these two are on the opposite side of the 6-membered ring, then the carbon 1 is said to be an alpha anomer. But if they are on the same side, the carbon 1 is said to be beta anomer. Alright, so this ring structure, the 6-membered ring structure which is formed, it depends on the arrangement of these two. That is the OH group, the OH group and the CH2OH group. Just remember that if they are on the opposite side, then it is an alpha form. But if they are on the same side, then it is beta form. So let us just take a look at this figure here. You can see this is a straight chain, okay, a straight chain, a linear form of glucose, but in the solution they occur in either an alpha form or a beta form. Now, I had mentioned just now that when the OH and the CH2OH group, they are on the opposite side, as you can see in this case, this one is in that plane, this one is in this plane. So if they are in the opposite side, it will be an alpha glucopyranose. Pyranose, since it is a pyranose ring here, a 6-membered ring, it is referred to as pyranose. So this is an alpha glucose, okay, alpha. Why? Because they are on the opposite side. But here you can see the OH group and the CH2OH group, they are on the same side. So since they are on the same side, they are beta form. Okay, this is a beta form. Okay, so if you see here in this figure, alright, so this is on the opposite side, it is an alpha form, and this is on the same side, so it is beta form. And one more thing I want to mention, this carbon number 1 here, okay, you see this carbon number 1 here, it is known as the anomeric carbon atom. This is the anomeric carbon, and these two forms, okay, in the beta D-glucopyranose and the alpha D-glucopyranose, they are referred to as the anomers. Okay, they are referred to as anomers. So next, I just want to talk about this hemiacetal and hemiketel formation. Now, an aldehyde, it can react with an alcohol to form a hemiacetal, and a ketone, as you can see here, this is an aldehydic group. Alright, an aldehyde, when it reacts with alcohol, it will lead to the formation of hemiacetal, and a ketone, when it reacts with alcohol, it will lead to the formation of hemiketel. Okay, so these hemiacetals and hemiketels, they are formed only on acidic conditions. acidic conditions when an aldehyde or a ketone they react with an alcohol respectively. And this reaction it occurs when a nucleophilic hydroxyl group, when a nucleophilic hydroxyl group of an alcohol it attacks the carbonyl carbon here. It attacks the carbonyl carbon of an aldehyde or the carbonyl carbon of a ketone. When this OH group, alright, the nucleophilic hydroxyl group, it attacks this carbon atom, or in case of ketone also this nucleophilic hydroxyl group, it attacks the carbonyl carbon of a ketone functional group, they will lead to the formation of hemiacetyl and hemiketals. And in the case of glucose, alright, in case of glucose molecule here, specifically this reaction it occurs intramolecularly between the OH group here, OH group of carbon 5, you can see here, this is the OH group, this one of this carbon number 5 in glucose. The reaction it occurs between this one, that is OH group of carbon 5, and the aldehyde carbon here, this one aldehyde carbon, resulting, you can see here, alright, resulting in the formation of a pyranose ring. You can see this ring here. This is in Fisher's projection, but in case of Howard projection you can see it is this one. So here you can see that the carbon number 5, you can see this is 1, 2, 3, 4 and 5. So the carbon number 5 with this carbon number 1 here, alright, it forms a bond, between, the bond is formed between carbon number 5 and carbon number 1. So these are the two forms, that is the alpha and the beta form, the two isomeric form of glucose. So in this way glucose it is known as, it can exist in two different isomeric forms as you can see, and these two isomeric forms, that is alpha and beta, they are termed as the anomers. Alright, so here the glucose in this reaction, in the case of glucose here you can see the reaction it occurs intramolecularly between the OH group of the 5th carbon atom and the aldehyde carbon, that is the CHO group of this carbon number 1, resulting in the formation of a more stable 6-membered pyranose ring which is named after pyran. And in this process a new chiral center is formed at the former carbonyl carbon, which is now known as the anomeric carbon. So here in this case this carbon here and this carbon here is known as the anomeric carbon. Alright, this carbon and this carbon here is known as the anomeric carbon atom. Now coming to the next topic that I want to discuss is Muta rotation. Now Muta rotation, the word muta, it means change. So Muta rotation, it is actually the change in the optical rotation. Alright, suppose in a solution the change in the equilibrium between two anomers, between two anomeric forms, when they change, Muta rotation is the optical rotation because of the change in the equilibrium between these two isomeric forms, that is anomers, when the corresponding stereocenters interconvert. Here actually what happened, the cyclic sugars show Muta rotation as the alpha and the beta anomeric forms they interconvert. Okay, so in solution, this alpha carbon can be converted into beta form, or the beta anomeric form can convert into alpha form in solution. So just remember that cyclic sugars, they interconvert from one form to another form, and that is termed as Muta rotation. And the optical rotation of the solution, it depends on the optical rotation of each of these anomeric form, and also it depends on the ratio between these two in the solution. And this word Muta rotation, Muta rotation in these anomers was first discovered by a French chemist. Okay, was first discovered by a French chemist, de Brunford, in the year 1844. When he noticed that a specific rotation it occurs, a specific rotation of the aqueous sugar solution, it changes with time. Now what happened here? When a monosaccharide molecule, okay, when a monosaccharide sugar is dissolved in water, what happened when it is dissolved in water? The optical rotatory power of the solution, it gradually changes until it reaches a constant value. Actually, the optical or the specific rotation of this monosaccharide sugar is 112. Okay, a freshly prepared aqueous solution of alpha D glucose, for instance, it has a specific rotation of 112.2 degree. And when this solution was allowed to stand, the rotation falls to 52.5 degree. Okay, and it will remain constant at this value. Whereas a solution of beta D glucopyranose, okay, a fresh solution of beta glucose, it has a specific rotation of 18.7 or about 19 degree, we can say about 19 degree. Now this will go on increasing until it reaches a constant value of 52.5 degree after few days. Okay, so this change of rotation is termed as Muta rotation, as I had mentioned, that Muta meaning change. So this process of changing is termed as Muta rotation or changing rotation. And this final stage, it can be attained more quickly either by heating a solution or by adding some catalyst, which may be an acid or an alkali. And this change is why it is changing like this. Why from 112 it is changing to 52.5 degree, or from 18.7 to 52.5. This is because due to the conversion of some of the alpha D glucose to beta D glucose, or vice versa, alright, or from beta D glucose to alpha D glucose. So actually in a solution, these will constantly change from one form to another form, from alpha to beta or vice versa. And when an equilibrium stage is attained, that particular solution will contain 1/3 of alpha form and 2/3 of beta form, and actually a very small amount of linear form will also be present. So just remember, in a solution when the equilibrium is attained, both these form will be present and a small amount of linear form will also be present. Okay, about 2/3 of beta form will be present, 1/3 of alpha form is present, and small amount of linear form when the solution it reaches an equilibrium. And that in that case the rotation will be at 52.5 degrees. So this is only when the rotation, it reaches this one only in when the solution is in when these two forms are in equilibrium. Alright, so just you have to remember that important point, that in equilibrium, at equilibrium, each solution will contain 1/3 of alpha, 2/3 of beta, and a very small amount of linear form. But suppose if the equilibrium has not reached, then it is found that the alpha D glucose will have this specific rotation, which will decrease until it reaches a constant value. That means that in that case, some of the alpha form has changed into beta form. Same thing in case of beta form, you can see that a specific rotation is 18.7, but with time, some of the B form, they will convert into alpha form until the specific rotation changes from 18.7 to 52.5 until it reaches a stable value. When it reaches this stable value, that means that solution, it is at equilibrium. So what is the mechanism of this Muta rotation? Now the phenomenon of Muta rotation, it appears due to changes of these two forms, as I had already mentioned, from alpha to beta or from beta to alpha via straight chain aldo or keto form. Through that straight chain, we know that sugars in solution, they occur in cyclic form. So this cyclic form may be alpha or beta, but Muta rotation, it occurs due to the interconversion between these two forms, from alpha to beta or beta to alpha. And as I had mentioned that when the equilibrium is attained, the characteristic rotation constant value is reached. Now according to Lowry in 1925, Muta rotation, it can take place only in the presence of an amphiprotic solvent. What is an amphiprotic solvent? It is a solvent that can act both as an acid and a base. So only in the presence of such solvent, Muta rotation can occur. So it is assumed that now we know that water is such solvent, which can act as an acid and a base. And it is assumed that when Muta rotation takes place, what happened actually here? What happened actually when Muta rotation occur? This ring, opens and recloses, either in the inverted position or in the original position. So suppose when an alpha form is present in a solution, how Muta rotation takes place, it is when the ring opens and recloses. So it is either when it opens and recloses, it either takes an alpha form or a beta form again until the equilibrium is attained. So that is the mechanism of Muta rotation. Now let us just talk a little bit on this chiral carbon or asymmetric carbon atom. Now, and as I had mentioned that an asymmetric carbon atom, or also known as a chiral carbon, is a carbon that is attached to 4 different groups. 4 different types of atoms or groups of atoms. And for each chiral carbon in a molecule, there are 2 optical isomers. You need to understand this, that here you can see, for example, in this molecule, these are the chiral carbon. Why? Because if you take a look at this carbon, for example, this one, it is attached to this group, this group, this group, and this group. So this carbon atom, it is attached to 4 different groups. So it is a chiral carbon. Same thing with this one, and this one. If you take a look, they are attached to 4 different groups. So they are chiral carbon, or known as asymmetric carbon. And here just remember, for each chiral carbon in a molecule there are 2 optical isomers. Okay, so as you can see here that for each chiral carbon in a molecule there are 2 optical isomers. That means if there are 2 chiral carbon in a molecule there will be 2 optical isomers. So let us take a look at this one. For each of this chiral carbon in a molecule there are 2 optical isomers. So here there are 4 chiral carbon, so that means there will be 8 optical isomers of D-glucose. Since for each of this chiral carbon there are 2 optical isomers, so 2, 2, 2 and 2, it will be 8 optical isomers. This can be written as this, can be calculated by 2 to the power n, where it indicates the number of chiral carbon. Now just now we see that in glucose we have 4 chiral carbons, so it will be 2 to the power 4, that means 8 optical isomers. So that means this increases exponentially with more chiral carbons. When a molecule has 2 chiral centers, then there will be 4 optical isomers. If a molecule has 4 chiral centers, then it will have 8 optical isomers. Okay, so that means there are 8 optical isomers of this particular molecule which has 4 chiral centers, that is glucose, D-glucose molecule, and there are 2 ways where the atoms can be arranged at each chiral center. Okay, there are 2 ways. For example, here in this chiral center, this can be on this side, this can be on this side. Same thing you will see in different optical isomers, okay, of this particular glucose molecule. There will be a difference in the arrangement of these atoms, okay, of these groups as compared to these chiral centers. There will be 2 different ways, or 2 ways of arrangement, different arrangement. Suppose this can be written like this, and the other isomer, it can be differ in any one of these carbon atom. For example, here in carbon number 2, the OH is on the left and the H is on the right. In another molecule, the OH can be on the left and the H can be on the right. So that is why here you see it will lead to the formation of these optical isomers, as there are 2 ways where the atom can be arranged at each of these chiral centers.